Dr. Pier Pandolfi received his MD and PhD from the University of Perugia in Italy after studying philosophy at the University of Rome as an undergraduate. He completed postgraduate training at the National Institute for Medical Research and the University of London. Dr. Pandolfi joined Memorial Sloan Kettering Cancer Center in New York in 1995. Currently, he has long-tenured distinguished professorship at the University of Turin and scientific advisor of the National Research Council of Italy. Dr. Pandolfi.
Good morning, everyone, and thank you, Liz, for the introduction. It is, where did he go? There. And thank you to the organizers, Christine in particular, for this opportunity to be with you today and honor the memory of Caris Sang with science. Caris was a scientist, was a physician, and she really spent her whole life attempting to treat manifestations of PHTS and in cancer prevention. It's tragic, I would say, and ironical, the fact that she succumbed to cancer. Now, Caris and I were friends, personal friends. We got to know each other when we were babies, very young. I will never forget the first time I met her. She was we were assistant professor, what they say in academia, junior faculty. And I had organized a meeting. I was at Sloan Kettering at that time in Arizona. And I sit at the table, and here she is. And she was way younger. She was like here, like in this picture. So our friendship started and persisted throughout the years. And then we started collaborating heavily. This collaboration is still ongoing. And I have to say, and I will first of all discuss data which we obtain together, very important, and which are published, but there are still studies that need to be accomplished and finished. So we will push hard for her legacy to persist. So she, for sure, wanted us to do science in honor and her memory, nothing else. And so to this end and in this respect, let me understand how you go forward here. Is the mouse or the keyboard? The keyboard. Okay. So to honor her memory, I have to say that I'm glad to be here today because I have a very exciting story to share, a story which is full of surprises, and I will discuss some of them, some of the unpublished, and at the end, a story which has tremendous implication for the prevention and therapy of PHTS and cancer, a story which stems from our long-standing interest in PTEN and in particular the mechanism that control PTEN function and which, as I will show you, are actionable at this point. Now, these are my disclosures. I have nothing to disclose as far as this talk is concerned, but I would like to bring your attention to this little biotech called Harbor Health, and I will tell you in due course why we believe that this biotech could be useful to the mission. Now, the protagonist of the story is PTEN, which really doesn't need introduction in this audience. However, I will reintroduce it to you because I would like to bring to your attention a few aspects which are relevant to what I will discuss today. Now, PTEN is by now one of the real gods of tumor suppression. In cancer is perhaps, along with P53 that Dr. Tramontana mentioned, the most mutated, silenced, deleted, functionally inactivated tumor suppressor in the history of cancer genetics. So I would like to say to the patients and the family, I say it all the time, that the interest on this gene is paramount. And therefore, you are not alone, and you will never be alone because the community of cancer people also is pushing very hard to study PTEN. PTEN is a phosphatase has many functions which are tumor suppressive, but the most important and pivotal for the discussion is that it can dephosphorylate as a lipid phosphatase lipid at plasma membrane. This blue stuff here is the membrane of our cells, and there are lipids that stick out, and what PTEN does, it dephosphorylates this lipid called PIP3 to PIP2, and in so doing, it's catalyzing the opposite reaction of a very important kinase, which serves the opposite biochemical function. It phosphorylates PIP2 to PIP3. So there are these two guys at play, P10 dephosphorylating this lipid to this lipid, and this kinase phosphorylating this lipid to this lipid. So if P10 is missing or functionally impaired, even just a bit, this leads to the accumulation of this product, PIP3, and this in turn unleashes a signal transduction pathway, which bestows cells with a number of proto-oncogenic properties. The cells start growing, they become motile, they can move around, and so in cancer they can metastasize, they can metabolically rewire, they acquire stem cell function and properties, and then at the bitter end they can lead to neoplastic transformation. Now, Dr. Tramontana already mentioned that obviously P10 is not only involved in cancer, but is involved in a number of syndromes which he described. They are all now referred to as PHTS, as we all know, because they are characterized invariably by two distinctive features: developmental defects, hamartomas, and cancer susceptibility. Now, the reason why I'm showing you this slide is not to remind you what you all know, but to bring to your attention the fact that these mutations are always found in heterozygosity, which in jargon means that when you have a syndrome, you still have a normal P10 allele, a normal P10 gene in your body. And this is very relevant because this normal P10 allele can become the target of therapies, and I will show you how. The other thing that I would like to remind you all that these syndromes are associated with P10 mutation, but not always. You can see these frequencies here. They are not 100%. And this means that there are other things out, other genes that we don't know of that can cause PHTS. And this was the focus of the work with Caris. And in fact, we did one major inroad and another is coming. Now, P10 is not only a very important, but a very special tumor suppressor, starting from the fact that it's a haploinsufficient, bona fide haploinsufficient tumor suppressor. What does this mean in jargon? It means that if you lose one allele of P10, you are already experiencing all the things I showed you in the previous slide. So the cells are growing, moving around. You don't need to lose completely P10 for this to happen. And this is different from P53, where P53 is technically not haploinsufficient. So you need to lose, although in some tissues might be haploinsufficient as well, you need to lose both alleles of P53 for an outcome to be manifested. And what we also discover, and this is really important, is that P10 is not only haploinsufficient, but is an obligate haploinsufficient tumor suppressor. What does this mean? Is that the cells cannot afford to lose P10 completely, because if they do so, they would be put out of business by a very important fail-safe mechanism, cellular senescence, which simply put impede the cells from proliferating further. So they are out of the game. So this is very important, and by the way, explains why you cannot be born without P10, because if you are born without P10, you would be experiencing a massive senescence response, and this is the reason why P10 is not, complete P10 loss is not compatible with adult life. But this in turn means that cancer cells love to lose P10 a bit, but they cannot afford to lose P10 completely. And again, there is still an allele, a gene of P10 to act upon, because P10 is very rarely completely lost, either in cancer and in PHTS. Now, the last point whereby P10 is very important is that those fluctuations in the up or in the down direction are very consequential in the case of P10. And I'm talking about subtle fluctuation. So these became very apparent when we studied in parallel two so-called allelic series. Simply put, what we did using genetic engineering is we generated mice which would go around the cage with 100% P10, so wild type dose of P10, 70% P10, only 30% reduction, 50% P10, which is what you would experience in PHTS, or 30% P10. Never zero, because zero is incompatible with adult life. And so by looking at these mice prospectively throughout their lives, some mind-boggling feature became apparent. The first is that if you drop P10 or just 30%, you would already put these mice at risk of developing cancer of multiple histology. And this is mind-boggling because it tells you that if anything kicks in, an environmental factor, a dietary factor, or a modifier, you could become at risk of developing cancer even if you don't lose P10 at all, so you wouldn't even see it. Now, the other important conclusion was that if you go down in dose, it becomes worse and worse. So if you go from 70 to 50, the tumor onset and incidence would go up. And if you go from 50 to 30, it would go further up and the tumor would become much more malignant. And why this is really relevant for the people in this room is that you are born at 50, but what you do with your life could bring you at 30 or back to 100. And I will come back to this during the talk. So there is yet another reason. This might be for you not that relevant, but you know that immune therapy are coming of age. And another important discovery was made in 2016, now confirmed by a number of papers, that is that if you lose P10, you become resistant to immune therapies. And this is important because, again, if you develop cancer in the setting of PHTS, you might be offered immune therapies, but you have to keep in mind that loss of P10 makes you resistant. And so, yeah, there is another reason why to go down in P10 is not a good idea. But the good news is that if you go up in P10, it's good at all levels. And this is also very important. So we made another allelic series, which is now creating mice that are higher in P10. And we could do that through genetic engineering. So we made mice that have an extra copy of P10, or 2 or 3 or 4 or 5, up to 7. So these mice are born with multiple P10 genes. And what we discover is that these mice are developmentally normal, and they are, however, super. We call them super P10 in many respects. They are long-lived, so elevation in P10 extends lifespan. They are lean, resistant to obesity, and they are, importantly, tumor resistant. So going down is bad, but going up is good. And this tells you that if we could find a way to super activate P10, even in people who have 2 normal copies of P10, this could impact human health at multiple levels. So my lab was interested in find a way to elevate, trigger, and reactivate P10 function for a number of years. And we believe that now we really cracked the code. We have 2 alternative ways to go after that. One, which I will not discuss today, which is this dimension here. Now, P10 is kept in check and in negative check by a number of little RNAs called microRNAs. These are, by definition, all proto-oncogenic, because if they keep P10 down, you would be at risk of developing cancer. And these guys can be taken out of business using decoys, small molecules that block their function. So to make a long story short, we went after one of them, which is called MIR22, which is a very nasty oncogene. And now we have a drug to go after this guy, a drug that I hope will be soon FDA approved, and that will be allowing us to reactivate PTEN. So in the years to come, we may end up having another way to reactivate PTEN. But today, I will discuss another dimension, which is more advanced, perhaps, which is laying its foundation on the identification of a molecular switch, which is really like an on-off switch. Imagine the switch of the light. And this is the on-off reversible switch which controls PTEN function at plasma membrane. Now, I told you that PTEN is there to dephosphorylate lipid. We have identified an enzyme called WWP1, which when active, simply puts this long chain of ubiquitin of the K27 type on PTEN. As a result, because of steric hindrance, which is to say because of this bulky adducts here, PTEN cannot accumulate at plasma membrane, cannot dimerize a plasma membrane. You can physically visualize PTEN falling off. And when PTEN falls off, then this enzyme that I told you before, P3K, kicks in, and its function is unperturbed, unabated, because the negative break is gone. So this is scary, but the good news is that this is reversible. So if you inhibit the activity of WWP1 genetically or pharmacologically, everything goes back to normal. PTEN goes back to the membrane, and it starts opposing P3K. So it's really on and off, is a switch. And what is the switch? This guy, WWP1, which has an enzymatic activity which is druggable. So when we discovered the switch, we jumped immediately on the chair for 3 reasons. The first is that it was already known that this guy could be an oncogene. It was found frequently overexpressed, amplified, genetically amplified, and mutated in many types of cancer. And in all these cancer, when WWP1 is in the hyperdrive mood, PTEN is down. So this explains how another mechanism whereby PTEN can be inactivated in cancer. But the reason why we jumped on the chair is that we immediately realized that this enzyme is druggable. Now, I don't know if this cartoon makes anything to you or means anything to you, but this is the crystal structure of the catalytic pocket of this guy. And you can see that in the middle there is a hole, a deep hole. This hole is druggable, meaning that you can hope to stick into the pocket a small molecule to interfere with the activity of this enzyme, hence reactivating PTEN function. And so to make a long story short, and you probably know already in part this story because it's published, we did find a potent WWP1 inhibitor from a cruciferous called indole-3 carbinol or I3C. So if you don't know what cruciferous are, and actually when I started, I didn't know what the heck cruciferous were. Also because if you look at the cruciferous, you ask yourself, why would they be called like the cross? There is no shape here. They are big, round, and these are broccoli, cauliflower, collard green, kale. But the reason why they are called cruciferous is because the flowers of these guys, it's always shaped like the cross, okay? So I didn't know that. That was cool. But that I started thinking, okay, this is really cool because your grandma, or my grandma, at least in Italy, would tell me to eat all this stuff because they are good for you. And so she was right, maybe. So this is, by the way, I3C in action. This is real science. It's binding to WWP1 at very low dose, 450 nanomoles. But what we didn't know is that you can buy I3C at CVS. You can buy it now, as these guys at Amazon. And I also didn't know that without doing any science, people had concluded that I3C is good for life extension. And I spent 2 hours of my life laughing because I couldn't believe that this guy had reached a conclusion which is accurate. Because if I3C inhibits WW and WW reactivates PTEN, I told you before that if you make a mouse that has more P10, it lives longer. So they might have been right without even doing the science. Now, seriously speaking, this is I3C in the pocket. We could model I3C in the deep hole of WW, and as you can see, it fits perfectly well. And with this information, we could generate a mutant of WW that is impermeable to I3C, demonstrating that I3C works, at least in part, by inhibiting WW, because if you make such a mutant, the cells don't respond to I3C anymore. Now, you might say, this is cool, but would it work? And it does work big time, both in vitro, I spare you the data, we fed cells with this I3C and they stop growing and blah, blah, blah. But importantly, it does work in vivo. We brought the I3C to the mouse hospital, which is still in place and where we test drugs in mice. And we enrolled two different models of cancer and one model of PHTS. So a prostate cancer model driven by the proto-oncogene MYC and a mouse model of PHTS, which co-develop a number of tumors. We let the mice age like human beings, they age and they develop cancer. And then we started feeding them with this dose of I3C, which is very well tolerated through IP injection for 1 month. And then we compared the mice fed with the placebo with the mice fed with the drug. And to make a long story short, the mice with the drug were tumor-free after 1 month. So this got published in Science, was discussed in a number of scientific journals, was discussed in a number of television, in a number of international and national newspapers. I have to say I'm very proud because I convinced the editor of Science to put the broccoli in the summary cartoon. So everybody discussed this study taking a very simple-minded angle, which is we found the reason why broccoli are good for you. But one reporter, Gina Colata, as you might remember, took a different angle. She said, and she was very shrewd actually, she caused a lot of problems in the community because she raised the question, what do you do now that you know that there is in the drugstore something that might be good for you? Would you buy it and take it on a daily basis? Or would you wait for a better drug? Would you wait for a more pure compound? Because when you buy it at CVS, you don't know what the heck you are buying. And so, by the way, this is the picture from that article. And you might recognize the people in this picture. But the mom is cooking broccoli for her daughter. The problem is that if you want to eat enough broccoli to have the dose that we gave to mice, you would need to basically eat 10 kilograms a day of broccoli, which is not that practical. So the debate ensued, and we finally found, or I found a way, which is that I convinced a group of young entrepreneurs to engage in the production of I3C in a pharmacological grade pure manner, which is pills that are gastroprotected, by the way, because gastroprotection is important for the stability of the compound and to avoid any intestinal side effects. And this compound is now available for clinical trial for free. So basically, if we want to engage, at least we know that we can use this guy, which is pure and GMP grade. Now, I'm almost done, but I told you this story is full of surprises, and this is, again, to honor the memory of Caris. So we started thinking that if WWP1 is a negative regulator of P10, and WWP1 superactive would be equal to P10 down, could it be that WW would be mutated in the germline in PHTS? And I told you that this was a legitimate question because there are a number of PHTS individuals that do not harbor P10 mutation. And so to make a long story short, we did find that this is the case. We published in the New England WWP1 gain of function mutation inactivate P10 in PHTS and are also found in the germline of individual with cancer, making WWP1 one of the most exciting new cancer susceptibility genes out of there. But the story is full of surprise, and now I close by giving you two suggestions for possible clinical trial. Now, the other thing that my lab discovered is that WWP1 inactivation, or I3C as a drug, would synergize with PI3K inhibitors. That was, in a sense, obvious, because on the one hand, you are reactivating P10, on the other hand, you are killing the PI3K enzyme. So the synergy was absolutely predictable. What was not predictable is that what we discovered is that I3C lowers one of the major toxicities of PI3K inhibitor, which is diabetes. If you take PI3K inhibitors, which could be taken even in PHTS, you have a big problem, clinical problem, which is that pseudo-diabetes ensues because PI3K is fundamental for glucose utilization. And the surprise was that if you treat combining PI3K with the I3C, the glycemia would be normalized through a completely independent mechanism, which is the reactivation of this enzyme in the muscle. So when you give I3C, the muscle starts sucking in glucose, and at that point, the glycemia is normalized. So this is very important because it suggests that in the future, we could aim at clinical trial with I3C and low-dose PI3K inhibitors, both in cancer and PHTS. The last clinical suggestion stems from the teaser I gave you before. I told you that if P10 is down, you become resistant to checkpoint inhibitors. So would I3C boost P10 and hence potentiate the efficacy of PI3K inhibitors? And the answer is absolutely yes. This is a very striking result. So I3C potently synergizes with checkpoint inhibitors. One, we tested ipilimumab. This is a colon cancer model in mice which are immune competent. If you treat them with an IgG, they die very fast. If you treat them with a checkpoint inhibitor, they live longer, the blue line. If you treat them with I3C, they also live longer because I3C is a tumor suppressive drug. But if you combine, they live much longer. And in summary, and I'm done, we found a way to resurrect and superactivate P10 to suppress and prevent PHTS manifestation, tumor regenesis, and to boost cancer immune therapy. And all these rest on these cruciferous and broccoli, which basically put you in a super P10 state, halting PHTS manifestation and cancer. These are the people who did the work, some of the people, I would say, the previous members of the P10 Dream Team and the current member, Maria Laura Sierra Cox, Chelsea Cox, Scott Sachs, and Ivan Neveu. We have been helped by Winnie Wei for a long time in studying E3 ligases, Julie Terria-Felsin for pathology, Sean Cloesy, the director of the Mouse Hospital, Lydia Matesic for the WW knockout, Massimo Bonora, Paolo Pinton, Giuseppe Novelli, and Federica for work that I have not shown you. And finally, Caris and Lami Sieia, with whom we have collaborated and we are still collaborating. Again, I would like to dedicate this talk to Caris and Caris' memory. Thank you very much for your attention.
We're going to take 2 questions, but I want to let everybody know that we're going to do a doctor panel at the end. So we're going to ask more questions at that point in time. Does someone have a question? Yes, Jimmy. Stand up so we can hear you well.
Just to wrap up your talk, if you have a child that has PGN, whether it's a young child or semi-adult, are you recommending that we go out? I know that Bracamax is treated in Europe as one of the frontline treatments for cancer, but on the ones that you just put up, is your recommendation that that be added to the child's protocol?
I do, but
Daily addition or what?
I do, but I think we really need to do serious clinical trials, and we are poised to do that. And we can discuss as a community actually where to start, because the one thing you don't want is to do something that is such a long-term effort that you will never know the answer. So when you do a smart trial, you need to gear it to assessing something that is measurable in the short term. And so we have been discussing about dermatological lesions. We have been discussing about colon polyps, thyroid nodules. If you were to take the drug systemically or per os, if you were to take the cream, perhaps we can do derm to start with. So we are open to anything, but I don't think it's wise to simply take the pill, because you will never be able to know if we don't design the trial in a way that we have some sense that this is working. Of course, I would take it. I by the way, I'm taking it because every day, because the other thing we publish and discover is that this I3C is also a very cool antiviral drug. So it worked beautifully. We found, in a sense, during COVID, we were stranded, so we couldn't do anything. We found that I3C works. We have mechanism in RNA viruses such as COVID and the flu. So I'm taking it. But would I know if later on I develop cancer or not if it was I3C? The only way is clinical trials.
One more question.
Would taking I3C now and the pharmacological dosage, would that cause problems being a research participant in the future? Would that be a?
Yeah, I don't. It's a good point, and actually, probably, But we have with Caris, we have been discussing about these possible trials, and then she got worse. Now we have to resume, and we have to do it fast because otherwise people will start taking it. And
Does it leave your system quickly?
yeah, no, it's a drug that is catabolized so rapidly. So very possibly the clinician would not argue if you were taking it. But I would take not that I'm here. I have a conflict because you can buy the Harbor Health compound. Try to buy something that is decent, because if you don't know what they put in these pills. And by the way, many are not gastroprotected, so you will not be pleased. But
Thank you.
Thank you.
Thank you very much, Dr. Pandolfi.